matrix is just the bacterial cytoplasm, ancestral bacterial cytoplasm. And in transit, it has to do a pit stop. So before it gets into the mitochondrial matrix, it has to do a pit stop at this thing called a PDH. What does PDH stand for?
Pyruvate dehydrogenase complex.
Let me draw it this way. I'm not going to draw it this way. Yeah, pyruvate dehydrogenase complex, right? Sometimes called PDC or PDHC, right? So let me do this. Let me put a, there we go. So PDH complex. Yeah, it's not as abbreviated just as PDH or PDC. It doesn't really matter. Okay. Not super high yield. So let me paste the complex here. So we got pyruvate that comes in, right? This is the structure of the PDC or PDH complex, pyruvate dehydrogenase complex. There's a lot of different subunits here. We got some acyl dehydrogenase. We got a bunch of different enzymes. Guys, if you could mute yourself, that'd be great. Less distracting. Thank you very much. So what do you know for the MCAT for the PDH step? Well, just like in glycolysis, we need to know the output. What's the output of PDH complex? What's the output?
I think it's what's written in red, so Acetyl Coa.
Okay. What else?
Maybe NADH H+. Okay.
So remember, right? How many pyruvates do we actually make per sugar? Make 2, right? 2. So maybe I annotated that on here as well. So when we get to this step right here in yellow, right? Everything after this step becomes 2 times, right? So maybe what we'll do is we'll do a little dotted line here, dash line. Sound effects are important. And we just have 2 of everything, right? 2x. Maybe I'll put 2x really big and bold here. Maybe you can even make it cooler, right? 2x, I don't know. Make it nice. Maybe we'll make it thicker. Let's do that. Let's make it really thick. 2x, 2 times the fun. Okay, so we have 2 times the fun with bubble mint gum. And so that means we have 2 pyruvates, right? We got 2 pyruvates coming in. And so it's not just one, it's not just acetyl CoA or NADH, it's specifically 2, right? 2 NADH. 2 NADH. Okay, and then I think the acetyl CoA is kind of implied as an output because that's what goes into the Krebs cycle, right? And where is this? What is the location of the PDH complex? Do that in dark green to be consistent. Where is that found at? Where's the PDH complex? Inner membrane. The inner membrane of the mitochondria, right? So it's going to be the inner membrane of the mitochondria. It's going to be attached to the inner membrane, right? Has a lot of different protein machinery and complexes. Tethered proteins are typically they're tethered to a membrane. In this case, is the inner membrane of the mitochondria. And then once we have the acetyl CoA, and maybe, I don't know, should we try to orientate this? Can we switch this? Can we kind of program, please work? Does it allow us to rotate it? It doesn't. That's all right. I know it's not that cool of a program, but that's all right. So we got the acetyl CoA, and that's going to be transported from the inner membrane. And we'll talk about shuttles later in one of the questions. Go ahead. Hello? Never mind. All right. So this acetyl CoA, right? It's going to interact with the Krebs cycle. Okay. So we have the acetyl CoA as a starting material for the Krebs cycle, right? And we're going to start getting into some tips and tricks here just in a moment, guys. That'll be nice. Finally. So we got acetyl CoA. And where's this acetyl CoA? Where is the Krebs cycle found? What part? about the mitochondrial matrix? Mitochondrial matrix. Right? Another way to say this is this, right? If we think about the endosymbiotic theory, right? What does the endosymbiotic theory say? Endosymbiotic relationship theory. What does it say?
Isn't it something about like mitochondria and bacteria or something?
Yeah, mitochondria are basically bacteria, right? Ancestral bacteria, right? So at some point, right, eukaryotic cells ate up, engulfed a bacteria that could do aerobic respiration, right? So the eukaryotic cell itself, right, it could undergo metabolism, but the metabolism that the cell did was glycolysis, which is anaerobic, right? There's no oxygen in glycolysis, right? And that's in the cytoplasm of the cell, of the eukaryote. But once it engulfed the aerobic respirating bacteria, okay, it can now take oxygen and use it for metabolism. So basically enslave this bacteria, this poor bacteria that's been enslaved, shackled inside of this eukaryotic cell to do hard work. Hard work, right? So it's done that, and now all we've done, confusingly, is called the bacterial cytoplasm, where the bacteria, right? Bacteria is just a bag of soup, bag of enzymes, bag of proteins, right? So this mitochondrial matrix is just code for bacterial cytoplasm, right? So if you took the mitochondria out, the Krebs cycle happens within its cytoplasm, which we call the mitochondrial matrix. Okay, cool. So Krebs cycle. We're not going to draw the Krebs cycle, but I will give you guys a pointer how to remember at least some of the structures quite easily. Okay, hopefully, I think that's that was one of the struggles I had, how to memorize some of this stuff. And Andrew, are you still here? Andrew Lee?
I'm here.
Yeah, you told me a good little mnemonic to remember the enzymes. Do you remember what that was?
So at disco.
Yeah, we'll talk about that here in a sec because I did like it, and I did steal it from you. So kudos to you.
Hey.
So maybe I'll pick on you in a moment. Okay, so we got this acetyl CoA as a starting product, and the acetyl CoA is going to combine with citrate, or excuse me, oxaloacetate to make citrate. Yeah. So acetyl CoA comes in, combines with oxaloacetate to make citrate. You guys know that step, right? Okay. So everybody has a little different mnemonics. One of the mnemonics that people use to remember the substrates has to do with selling drugs. Which mnemonic is that? Something like about, officer, can I keep selling drugs or something like that? You guys remember what that is? That mnemonic?
I remember it as, can I keep selling sex for money, officer?
Oh, it's sex, not drugs. I'm talking about, can I keep selling sex? What was it?
For money, officer.
For money, officer. There you go. Hey, I can tell you to spend your time. OnlyFans. Can I keep selling sex for money, officer? Right? So that's one that people quite use. That's very popular. The one I came up with is, can I ask sexy Susan for my orgasm? For my orgasm. Since I'm a guy, right? Okay, that makes sense for me. If you're a woman, maybe replace sexy Susan with sexy Steven. And if Steven's any good, it's multiple orgasms, right? Can I ask sexy Steven for my orgasms? Okay, so pick one of these that you'll remember. Okay, it's going to help you remember the substrates. It's a little bit perverse. I make no apologies for it. Okay, but it's going to help us remember the substrates of the Krebs cycle, and that's important as well. Now, students sometimes have issues remembering whether succinyl-CoA or succinate is first, right? Because we have two S's in line. Selling sex or sexy Susan, it doesn't really tell you whether you have CoA first or succinate first, right? Here's a tip. Succinyl-CoA is going to be first. and then it's going to be succinate. How can you remember that? Well, if you think back about acetyl CoA, right? If you think about acetyl CoA, it was the CoA that's first, right? Acetyl CoA is first. Therefore, succinyl CoA has to be first. All right. So let's talk about the enzymes. Another mnemonic that's really helpful that Andrew shared with me about 2 weeks ago, 3 weeks ago, a month ago. And it basically was this. Remember the enzymes, and we'll write down the enzymes here in just a moment. It's I think it was disco, devil, sipped, down, 5 drinks. A lot of D's here, right? Disco, right? Disco, devil sipped down 5 drinks. I don't know if you guys knew this, but the devil is a disco fan. Staying alive. Staying alive. So at disco, devil sipped down 5 drinks. Okay. So with that in mind, okay, the SO refers to, let's see here. Let's see, I'm going to write this. Maybe I should have done this in different colors here, but maybe I'll do this I guess we do this in black to be consistent with it. So that the first enzyme from oxaloacetate to citrate, any guess on what that's going to be?
Citrate synthase.
Yeah, it's going to be synthase comes into play, not just any synthase. So that's SO is for synthase. It's going to be citrate. Citrate synthase. Love the enthusiasm. Yeah. Okay. And then if we go from citrate to isocitrate, what do we got there? Starts with an A, right? So that's the at.
Conatase.
Ooh, nice. Conatase. And next step, well, it starts with it's not necessarily starts with a D, but the D refers to a dehydrogenase. We also know it's a dehydrogenase because, right, we get NAD to NADH, and that requires a dehydrogenase to removal of a hydrogen to NADH, or NAD to make NADH, right? So that's a dehydrogenase. It's a telltale sign we're dealing with dehydrogenases. So that's the D, disco for dehydrogenase. Let's write that. But it's not just any dehydrogenase. What is it?
Isocitrate.
Yeah, so most of these, not always, unfortunately, but most of the times when in doubt, just look at the substrate of the product and it's going to be one of the two. So in this case, right, a lot of enzymes will take their name after the substrate of the product. So in this case, it's going to be isocitrate. And then what does it do? Well, it removes a hydrogen, so it's going to be a dehydrogenase, right? Simple enough. Awesome. All right, so we got the we got the disco. Golly, but now got the devil. Son of a gun. Devil went down to Georgia. So that's also going to be dehydrogenase, right? You see the NADH, that's a dehydrogenase, hence the D for devil. Let's write that out. Dehydrogenase. All right. So based on the naming convention we just talked about, what most likely is the first name, or what's the name of this dehydrogenase?
Alpha-ketoglutarate.
Yeah, it takes its name after the substrate typically. We'll see there's going to be some exceptions here in a moment. Alpha-ketoglutarate. And I'm going to send you guys a sweet little Krebs Cycle rap video. So I used to go to the gym. That's another thing I did too. I remember it was before COVID hit, right? I went to the gym and listened to this Metabolism rap song, and I'd be jamming out to it, getting all swole, both from lifting and getting my brain swollen from that rap song. Okay, it's another way you can cheese it. Not proud of it. It's not the proudest moment of my life, but you got to do what you got to do. All right. So next step, right? We're making some GTP, and GTP in terms of the MCAT. Just assume that's equivalent to ATP, can be converted to ATP directly, okay? So we got the S, we got the at, we got the disco, we got the devil. That was the alpha-ketoglutarate dehydrogenase. Next thing, we've got S. So that's going to help us out a little bit. So S, like before, right? So S was what before? Synthase, right? So in this case, the S here is also a synthase, right? We're synthesizing, making something. In this case, is GTP or ATP. So let's put that down. Sipped for synthase. And what kind of synthase are we dealing with? Anybody?
Acetyl Coa.
Yeah, succinyl CoA synthase, right? Takes its name from the substrate. Too easy. My point is this, kind of, the reason why we're doing this, right, is that this shouldn't be that hard, right? If you understand the naming convention of enzymes and you understand what's happening, too easy, yeah? So this mnemonic, so at disco, double sip down 5 drinks, is just a way to help you out, right? Cool. So then we got succinate going to fumarates. What do we notice here, though? We got FAD becoming protonated. So we look at the word sip down, and where it's getting protonated. What kind of enzyme do we have?
Succinate dehydrogenase.
Killing it. You're a beast. Nice. Succinate dehydrogenase. Yeah. Dehydrogenase. Too easy. Cool. And then the next one is a little bit weird. The 5 drinks 5. This is actually going to be fumarase. Right. And then we've got the last step is going to be this drinks, right? The D, every time you see D, you should think of dehydrogenase, right? And that's what the step shows here as well. So we don't have just any dehydrogenase. We have malate dehydrogenase. Cool. So again, I think any combination, if you know 2 of these or all 3, I think you'll have a good understanding about the order of the substrates as well as the enzymes. Can I keep selling sex for money, officer? Or can I ask sexy Susan or Steven for my orgasms, whatever. And if you know that, you know all the substrates, one of the 2. And if you know the one in black, so at disco, right? The devil sit down 5 drinks, devil, right? Not only does he like to go down to Georgia, he also likes to dabble in disco. Okay. If you know that, you know all the enzymes. Pretty easy, right? It makes this a little bit easier to understand. So we're going to talk about the output real quick, and then I'm going to give you guys a tip on how to rationalize some of these structures like fumarate, malate, and oxaloacetate. Make it a little bit easier. And I'm going to tell you guys right now, you guys already know it. You may not realize it, but you already know it. Okay. I'm just going to kind of open the gates there, be the gatekeeper to that. So you guys realize you guys have full potential. That's always nice. Okay. So, Krebs cycle. What's the output? What's the output of the Krebs cycle? And let's do it, the output for 2-pyruvate, right? For every glucose molecule, we have 2-pyruvate. So for every 2 pyruvates, what do we get? Pyruvates. I can't spell today. Let me do this again. Pyruvate. There we go. Bingo. Good job, Kevin. Thank you. What do we got? Well, let's look at this. We've got what? We got 1, 2, 3 NADH per pyruvate, but we're saying, hey, let's do that for 2 pyruvate. So instead of having 3 NADH, we double that. So we have 6 NADH. Yeah. Okay, cool. How many FADHs do we have, you guys, for 2 pyruvate?
2.
Nice. Love the enthusiasm. Okay. And then ATP, we also have 2. Cool.
Also, I think the FADH should be FADH2.
Oh, pardon me. Thank you. I like it. Absolutely correct. Stand corrected. Good job. Excellent. Yep. It is FADH2, not FADH. Excellent. All right. So let's do a quick little tangent and talk about how to cheese the structure for malate, fumarate, and oxaloacetate. Okay. Because I know this was a problem that I had when I was first studying this. I'm guessing it's going to be a struggle for you guys as well. So we're going to take a quick tangent. Okay, we're going to walk down the halls of Narnia for a moment. And specifically, we're going to talk about fermentation for a moment. Fermentation. And maybe I should find a picture on fermentation. Do this. I actually have one, luckily. And I don't know if it'll move further to the right or not. Let's see if I can get this program to squeech further to the right. So glycolysis, right? Pyruvate is produced under aerobic conditions, right? But fermentation essentially is glycolysis under anaerobic conditions, truly anaerobic, right? There's nothing downstream. There's no Krebs cycle. There's no ETC. It's been essentially cut off, or is just backlogged, right? And with fermentation, so let's write that down. So fermentation. This is going to be a good prelude. Fermentation is essentially when you've got glycolysis, or truly anaerobic. It's basically anaerobic glycolysis, right? There's nothing attaching it to aerobic respiration, glycolysis. All right, any questions on that so far? Hopefully I didn't confuse anybody. What is the purpose? Can you tell me what the purpose is of fermentation? Why does our body, why do we produce lactic acid, or why do we do that? What's the point behind that? Why is it that we turn pyruvate, right? So when we undergo fermentation, right? Because we're not breathing in enough oxygen when we're sprinting, working out, right? Getting our beach bods going on at the gym. Why is it that we get sore? Why is it? Well, I already know that's because lactic acid buildup in part. But why is it the body, right? If we look at this glucose fermentation pathway and look at glucose here, right? The glucose undergoes glycolysis and makes 2-pyruvate. We know that already. We just went over that. But then the next step in fermentation is it produces lactate. Why is that?
To regenerate the NAD+.
Yeah, regenerate NAD+. There's only so much NAD+ hanging out in the cytoplasm. There's only so much of it. Typically, the NAD+ is regenerated when it encounters the ETC. Right? So the NADH will drop off its protons at the ETC, right? But here, we don't have the ETC anymore. We don't have the Krebs cycle. It's been backlogged. It's been removed from the equation. So that's not a viable avenue anymore. So in order to regenerate the NAD+, that NADH, that extra proton that's carrying, that NADH, right, that proton it's carrying has to be dropped off. And it's going to protonate pyruvate and turn it into lactate or lactic acid. So you can regenerate that NAD+ to continuously do glycolysis, right? This happens with cancer cells too, right? Cancer cells do this quite a bit. There's a high turnover with glycolysis because it has such high energy demands, right? Because it's reproducing or replicating uncontrollably. Okay, cool. So why am I making a big stink about this? And how is this going to help us remember the structures for malate and oxaloacetate? What gives? Okay. Well, we'll get there. Promise. So this metabolism is for mammals typically, or eukaryotes in general. I guess yeast could be considered eukaryote, although it does have kind of bacterial-like attributes. But if you think about yeast, let's talk about yeast for a sec. Yeast. Yeast can also go through this, right? And yeast, instead of producing lactate, what does yeast produce? Right? So let me actually back up just a little bit. We use yeast to make bread, and we use yeast to make beer or alcohol, right? So what does yeast produce? What kind of alcohol does yeast produce?
Ethanol.
Yeah, it's ethanol, right? Not methanol. Don't drink methanol, guys. Okay, that'll make you blind. That's the wrong kind of alcohol. Don't do that. It produces methanol, or excuse me, ethanol. I get me all confused now. Ethanol, right? Ethanol. So when yeast undergoes fermentation, it doesn't produce lactic acid or lactate. It produces ethanol. Okay, so yeast produces ethanol. Cool. And has anybody ever made beer before, or wine, or mead, or anything like that? Has anybody done that before? Maybe in microbiology class, or maybe as just, as a homebody, home enthusiast, recreational hobbyist.
We made rum in organic chemistry.
You made rum? Did you use yeast to make rum?
Yeah.
Yeah. So you might remember that you put a plug on top of that bottle to allow for gases to escape. What gas is produced in fermentation with yeast?
CO2?
Yeah, CO2, right? So the CO2 can escape, right? This is the same reason, right, when you add yeast. Why do we add yeast to bread, you guys? Well, we add yeast to the bread, or what will become bread, right, the dough, as a levitating agent, right? It levitates, or it doesn't actually like make it levitate per se, but it helps it rise. And how does it help it rise? Well, the yeast, as it consumes the carbohydrates, it releases CO2. It also makes ethanol, right? That's that yeasty smell. That's because that's coming from the ethanol. And it's also producing CO2. That produces the quote unquote air bubbles in the bread, right? So it is not so thick. It makes it airy because of the CO2. Okay. I don't know about you guys, but I made some quarantine bread when, COVID hit because I thought I was going to die. Okay. But that's why you add yeast. Now, you don't always have to add yeast. That's fine. You can also add other stuff, neither here or there. But the point is this. If you know what yeast produces, if you know that yeast makes ethanol, alcohol, right? Because you like to go to the bar and drink, okay? And you bake, it makes CO2, then you know the structure of malate, of oxaloacetate, and so forth. Let's get into it. Let me prove it to you guys. So first off, let's draw the structure of pyruvate, I think, right? So we got pyruvate. And you guys already know what the structure of pyruvate is. You guys may not know it, but you do. And pyruvate looks like this. I have to think about it here, to intuit here based on what I know. So, okay, 1, 2, O minus, okay? So there's pyruvate. And guess what, guys? I don't memorize this. I can intuit it, okay? And this is how you'll do it. So based on the fact that we know, right, yeast, the alternative pathway for yeast, right? So yeast take pyruvate, they take this pyruvate and turn it into ethanol and CO2, right? Ethanol and CO2 instead of lactate. That's a different byproduct, right? There's different types of fermentation. One is lactic fermentation, the other one's ethanol fermentation, right? Yeast was the latter one. Well, essentially all we're doing is we're breaking down pyruvate into ethanol and CO2. So if we want to know what the structure is of pyruvate, all we got to do is do the reverse. We got to do the reverse. We got to bring ethanol and CO2 back together, and then we have pyruvate. Let me show you. So, right? So let's do this. Let's draw ethanol out. Ethanol. Ethanol is what? 1, 2. And then we've got the alcohol group. Okay? And then we got what? We got a CO2 group, right? The CO2 group is simply going to be a carbon double bond O to a single O minus, give or take, right? There's your CO2. Okay, and then you just got to combine it. And, right, make sure you oxidize. Here's a little trick, I guess. You just got to oxidize this OH group into a ketone group, right? Make sure you do that, oxidize. Make sure to oxidize the alcohol group to a carbonyl. Okay, done deal. So here's let me do this in different colors. Maybe say the CO2 group's in red. And so we'll say this group right here on the right-hand side is also in red. Do that a lot better. Okay, so there you go. You just added ethanol and CO2 and you got pyruvate, more or less. Or at least you got lactic acid, you keep the OH group. Okay. Or lactate or lactic acid. Okay. Okay, so with that in mind, let's look at malate. Let's go back to the structures real quick. Let's look at malate. What do you guys notice with malate? Well, essentially malate, let me zoom in here so everybody can see this. Zoom in real close. Perfect resolution. What do you guys notice? What do we got here? Well, malate is simply what? It's the CO2 group right here, another CO2 group right there, and then what do we have in the center? Well, in the center we've got what? Ethanol. So malate, you could think of this as what? Malate is simply a CO2 ethanol CO2 sandwich. How easy is that? How easy is that? Malate is just an ethanol carbon sandwich. You got carbon on each side, you draw ethanol, you're done. Done deal. Pretty easy. Pretty easy. Okay, so if you reduce, actually let's go the other way around. We know what about Krebs cycle? We know the Krebs cycle, right? Every step essentially is what? Oxidizing, right? As we go around, we're oxidizing, right? We're breaking those bonds, oxidizing them to extract energy, more or less, right? For the most part. Okay. So if we oxidize malate, if we oxidize this OH group, well, we get oxaloacetate, right? So we take this OH group and we oxidize it. We get oxaloacetate, right? Oxaloacetate is simply the same thing. It's just simply two CO2 groups and an ethanol, except the ethanol or the alcohol group has been oxidized to a ketone. If we go the other direction, right? If we want to go from malate to fumarate, we just got to reduce. And if we reduce, an OH group becomes water, right? OH2. And water is a what? Water is a good leaving group, right? Hence why we lose water right here. So by knowing malate is simply a CO2 ethanol sandwich, got CO2 on both sides, right? We can then intuit the structure of both fumarate and oxaloacetate. All you got to do is oxidize, reduce that OH group. Not too bad, right? And guess what? You guys already knew it. You guys go to the bar and drink alcohol, I'm sure. You've had alcohol once in your life. Okay. You're drinking ethanol. You know what yeast do, right? Yeast make alcohol for us. There's the ethanol. And we make bread, right? You guys had bread before. Makes that CO2. Okay. That's it. You guys already knew it. Simple as that. Okay. All right. So that was my little bit of wisdom there. Okay. Any questions on that so far? Was that helpful-ish?
Yes, thank you.
Okay. All right. So we got the ETC next. Electron transport chain. Any questions, comments, concerns, or insults? I take them all before we move on to the ETC. Can I ask Sexy Susan for my orgasm? So I disco devil, sip down 5 drinks. Devil's a heavy drinker. Okay. All right. So now that we've all these NADHs and FADHs, right, we need to put them into action. So let's do that. So here's the ETC. Let's make this a little bit bigger because I have a squint. Okay. So ETC is in the inner membrane of the mitochondria. Okay. And it's as such. My job is done. No, just kidding. All right, we got other stuff going on here. One thing I want to talk about is pH. So on the MCAT, you will probably see questions that will ask you about drugs that either poke holes in the membranes or they're called decouplers, right? There's different types of drugs that have different effects. What is the pH of the cell cytoplasm? Does anybody know? What is the pH of the cell cytoplasm?
Seven.
Yeah, it's around 7, yeah? Good, right? Just like our blood is around 7, right? Our body pH is around 7, 7.2, 7.4, right? It's around 7. Cool. Here's the next one that people have troubles with typically. So typically, I think everybody understands that the inner membrane space, right? The space here in the center where the protons are being pumped across from the mitochondrial matrix, right? Into the inner membrane space. People understand that it's going to be more acidic relative to the mitochondrial matrix. And I would agree because you're pumping in protons. What people typically have an issue with is understanding what the pH of the inner membrane space is. And in fact, the inner membrane space is also around a pH of 7. It's also around 7. Interesting, kind of weird, right? But the relationship between the inner membrane space and the mitochondrial matrix does hold true. The inner membrane space is more acidic than the mitochondrial matrix, okay? For obvious reasons that we talked about, right? Because it has more protons being pumped into it. And the pH of the mitochondrial matrix is around 8, right? 8 is more basic than 7, so it still holds true that the inner membrane space is more acidic, even at 7, relative to the pH of 8. So why am I bringing this up, right? Well, I just talked about these drugs being able to poke holes. So the MCAT asks you, right, what would happen? What would happen if you have a drug that pokes holes, right, pokes holes? Let me do some red here. What would happen if drug, let's say drug A, what would happen to the net movement of protons if drug A poked holes in the outer membrane of the mitochondria? What do you guys think?
It would screw up the concentration somehow.
Would it? Are you sure? What's the pH of the cytoplasm? What would you say the cytoplasm was?
7.
7. And the inner membrane space, the pH is 7 as well, around, yeah? So pH is a measurement of proton concentration, right? It's a negative log of the proton concentration. So if they're basically in equilibrium just anyway, would it really screw the concentrations of the proton gradient up? Probably not. Probably not. All right, this is why we're talking about it now. Okay? So there would be no net movement of protons, right? Because they're equal in concentration. Right? The pHs are the same between the cytoplasm and the inner membrane space. Cool. However, let's think about this though. What if, let's do a different color. I don't know. Let's do like, puke green, right? Puke green's nice. If we had, maybe another drug, let's call it drug B. I know, fancy names. I impress myself sometimes. Drug B. If we had drug B, right, instead of poking holes in the outer membrane of the mitochondria, in fact, poked holes in the inner membrane, what will be the net movement of protons? And this should be, I think, fairly obvious.
That's what I was getting confused. Yeah, it would screw up the gradient here, right? And that
It
would.
would. And in what direction? Which way would it go? Which way would the protons go?
Wouldn't they be stuck in like the matrix?
Stuck? I don't know if the word stuck is right, but yeah, I think you're on the right track. It's these protons, right? Bunch of these protons, right? Because the pH of 7 is more acidic than 8. That means that the inner membrane has more protons, right? Would go back down their concentration gradient from a lower pH or higher proton concentration to a lower proton concentration or a higher pH, right? They would travel down the concentration gradients back into the mitochondrial matrix. Okay. Now, for the MCAT, you'll probably see the latter. You'll see drug B more often talked about than A, right? And I'm sure you guys have seen some of that as well. You will probably be asked how many protons are transported across each complex. Okay? And this is simply 4 for complex 1, 0 protons for complex 2, 4 protons for complex 3, and 2 protons for complex 4. Okay? So 4, 0, 4, 2, 40, 42. Right? So 10 protons across complex 1 through 4 are transmitted from the mitochondrial matrix to the inner membrane space. 4, 0, 4, 2. Okay. If one question you might be asked too, and it's a bit niche, let's go back to drug B for a moment, right? We said, well, if drug B allows for protons to go back down their concentration gradients prematurely, right? Because this drug B might be everywhere, right? You might have drug B in all sorts of different spots along this this gradient, or along these complexes, right? Might be interspersed. Let's put another one on the right-hand side, right? Drug B, and then we got H plus coming over here, and it goes down as concentration gradient before it's supposed to, right? Because technically it's supposed to go down the ATP synthase, right? It's supposed to make some ATP. It's supposed to go down its concentration gradient, and then that can be used for work, right? It's kinetic energy. It goes down its concentration gradient. And when ATP synthase, the protein, uses that energy to do work, and stores that energy in the form of ATP, the biological currency, right? So now we're saying, okay, well, let's think about this. What about this? What would happen if the hydrogen comes down its concentration gradient too soon? What would happen to your ATP? Would it go up or down?
Down.
Yeah, I mean, that's obvious. Duh, Kevin. Duh. Right? It's not interacting with ATP synthase. Cool. Okay. What would happen to the O2 consumption, though? Would it go up or down, and why?
Wouldn't it go down since it's the final electron acceptor?
Interesting. Maybe. Somebody want to agree with that or disagree? Let's talk about it.
I think it will go up because you're not making more product.
Okay. Maybe Red, maybe if you're still on here, maybe you can make a poll for us.
Do
In the
you mean
room one?
O2 consumption, so the use of O2 or just the concentration of O2?
Let's make a poll on this real quick. See if I can do it. I'm slow on this. So would O2 in the study room one chat, let me make this question. Would O2, let's move this over here. Would O2 consumption go up or down? And then I think I just go up, down. I think that should work. There we go. AB. What do you guys think? Everybody kind of click on that real quick. Select
Wait, and this is if you have drug B. Is that the question again?
Yep, drug B. Yep. So if it's drug B, right, it allows for the protons to cross over prematurely. What would happen to O2 consumption? Would it go up or would it go down? So everybody click on study room 1 text channel right above this channel and hit A or B. Maybe I should have put C on here too. Here, we'll put C on here as well for the undecided. There we go. I'll put C. Oh, just kidding. Just kidding. C on here as well. All right. All right. So most of you guys are saying up. Maybe we have one person change their minds. It's okay. Stand firm. Right. It's fine. So A is correct. It'll go up. So let's talk about that real quick because I don't think it's that intuitive. Okay. So we have O2 consumption that goes up, up and away. Why? Well, right. In fact, O2, although it on this picture shows O2 is over here, right below complex 4. Really, it's O2 is all over the place, right? O2 is just all over the mitochondrial matrix, right? It's hanging out all over the place. So as soon as it encounters hydrogen, H plus, right, it's going to turn into water. And so the O2 consumption would go up because a lot of O2 is immediately being used up because there's so much hydrogens, right? There's so many hydrogens ready to react with oxygen. And so that oxygen is going to be used up left and right to make excess water, right? So our H2O consumption or H2O production, right, because O2 is being consumed, is also going to go up. Does that make sense?
Yes.
Okay. If you guys have troubles with that, not a problem. Again, I always stick around afterwards to answer any questions or clarifications. And if you do have a question, right, you can also just text me, DM me, PM me, I don't know what the lingo is anymore. Private message me, direct message me, or just put it in the chat. Okay. All right, so that's metabolism in a nutshell. We can do a couple things. We can talk about high yield metabolic structures, some other ones, or we can talk about fatty acids, or we can do a combination thereof, and then at the end, or we can do the next hour. If you guys say, hey, I'm bored with metabolism, over it, that's cool too. We can just jump into some psych-soc, high-yield psych-soc, right? Stretch our legs out, get a little more jazzed, excited about something else other than metabolism, something that's a little more high yield. What do you guys want to do?
Fatty acids.
Gross. Anybody else?
High yield psych-soc, please.
All right. So like I said, right? So if you guys can do the questions, if you guys scroll up, you guys can see my screen, right? I post a lot of questions here. Okay. I kind of came up with them last night quite rapidly, but I think it really covers a lot of the fatty acid synthesis. And so what I'll do right now, I guess, to keep everybody happy is I'll go over them in turn quickly. Okay. And then we'll jump into some psych-soc, because I promised a lot of students last week, or I guess the week before, I'd do high-yield psych-soc, and just never came to fruition. Okay. Again, fatty acid metabolism is super low yield, but let's go over these questions real quick. So, question 1, uno numero. If you guys haven't done these questions yet, go ahead and jump onto Study Room, the text channel, right? Scroll up just a little bit and select your answer choice. So the first question is, immediately after eating a meal rich in carbohydrates, sugars, right? Which of the following processes will occur by location? A, fatty acid oxidation at adipocytes, right? Adipocytes are fat cells. Okay, well, why would that be wrong? Well, you wouldn't want to oxidize fatty acids, right? If you're eating a lot of carbs, you want to store the carbs into fat, right? You don't want to oxidize, you want to make fatty acids. So that's the exact opposite of what you would expect. Cool. B, gluconeogenesis in the liver. Well, what is gluconeogenesis? Well, gluconeogenesis is the mobilization of glucose into the bloodstream, right? So that's the exact opposite we're trying to do. We're trying to get that sugar into the cells. We're trying to pack that extra carbohydrates into the cells, into glycogen, right, as reserves. So B is the exact opposite of what we want to do. So I don't know who picked on that. Mitochondria. Okay, so he's not here with us today, but that's why that would be wrong. And as everybody pointed out, it would be C. The answer will be C, glycogenesis, okay, production of glycogen. That's exactly what we want to do. We want to take that extra energy, right, in the form of carbohydrates or sugars, and store it in the form of glycogen in the liver. And that happens primarily in the liver. Okay, good, right? That's why when people drink like a lot of alcohol, right, you may have heard of non-alcoholic or alcoholic fatty liver syndrome, or non-alcoholic fatty liver, same idea. When people eat too many hamburgers, McDonald's, right, they get non-alcoholic fatty liver syndrome. That's because they're producing too much fat around the liver. Or if they're drinking too much alcohol, it'd be fatty liver syndrome for the same reasons, right? Too much fat around the liver. Either way, I digress. So D, glycogenolysis. That's also the exact opposite, right? We don't want to break down glycogen reserves. We want to produce glycogen. Again, so that's the exact opposite of what we want. So the answer for this first question, maybe I should number them, is going to be C. So everybody got that mostly correct. Nice job. Whoop, whoop. High five. Hopefully you guys can get ahead of me a little bit. If you guys haven't had a chance to look at these or answer them, maybe get ahead of just a little bit if you can. Fatty acid synthesis occurs in which location, right? So I think this is an important factoid you need to know. Fatty acid synthesis does occur in the cytoplasm or the cytosol, okay? So A is correct. And then that kind of leads us, I'd go through the other questions, but I think the next question kind of highlights what the other answers are. So which of the following locations does fatty acid oxidation occur? It's the exact opposite, right? So if I were to make a note card on Anki, right? And on that Anki card, what I would do, let me bring up mine real quick. And maybe you guys want to copy and paste these questions, right? If you're not sure about it, copy and paste them, right? Pull up your Anki, okay? Make a deck or a card. I don't know how you want to do that. Make a new deck, right? Maybe call it Metabolism. It's going to let me do it. It's not. I don't know why. We'll talk about it later, I guess, or try to figure out later. Make a deck, the front, maybe copy and paste this question. Where does fatty acid acid oxidation occur? And where does it occur? Anybody? Where does fatty acid? I can't talk today. Where does fatty acid oxidation occur? Any guesses?
Mitochondria.
Hey, nice. I don't know how you knew that. Excellent. Where else? Where else?
And peroxisomes.
Yeah, so there's actually 2 locations. There's actually a third location, but you won't be tested on that. The ER does a little bit of what's known as omega oxidation. That's beyond the MCAT, so don't stress about that. But for the purpose of the MCAT, you need to know 2 sites where fatty oxidation occurs, right? So your beta oxidation primarily occurs in the cytoplasm and the peroxisome. Why is there a difference? What does the peroxisome do or take that's different from the the mitochondria? I misspoke there. So fatty acid oxidation occurs in the mitochondria and the peroxisome. What is that difference? Why is there a difference? So the peroxisomes also do beta oxidation, a little bit of alpha oxidation too. You don't even know what that is on the MCAT, but they both do beta oxidation. The peroxisomes typically will oxidize or beta oxidize fatty acids that are complex or very long. You don't need to know how long, but if you are type A like me and you need to know everything, right? It's actually, I believe fatty acids that are 22 carbons in length or higher. Okay. And then the mitochondria just basically does anything lower or more simple beta oxidation. Cool. And then side what? Fatty acid synthesis, yeah? That was the first question. Okay, so the answer for this is going to be 1 and 2 or answer choice C. Most of you got that correct. Nice. Next one. Ooh, so I was trying to be hard on this one. I'll be honest. These biological shuttles, I'm not too high speed on, so maybe I want to lean on some of you guys, a little more high speed than I am on this. Fatty acids must pass through which shuttle in order to pass through the inner mitochondrial membrane in order to be beta oxidized? So one thing you should know is that the outer mitochondrial membrane is typically not a problem for a lot of biological processes, right? And that has to do with the fact that, in part, it's a little more receptive for transport. So you could probably argue because the pH compartments are similar-ish, I guess. Maybe that's a bad explanation. But it's typically, the challenge is basically all the time the inner mitochondrial membrane. It's a little more fixed, I guess. It's not as passive or what's the word I'm looking for? It's harder to transport things through the inner mitochondrial membrane. Let's just leave it at that. So here, you got to know your biological shuttles, right? Is that super high yield on MCAT? Probably not, but it's probably good to make a note card on these or maybe this question. And maybe write your own answer key for A, B, C, and D so you can remember them, okay? So let me ask you guys this. What do some of these shuttles do? What does let's start on the bottom here. Let's change the pace a little bit. What does the Malate Aspartate shuttle do? What does the Malate Aspartate shuttle do?
I think it converts oxaloacetate to malate so it can go through.
Yeah. I mean, pretty close. Yeah, I like that. So essentially what's happening there, why would you want the Malate Aspartate shuttle? Because the problem that you might encounter is that you could have NADH, right? So remember that NADH is produced in glycolysis, right? And glycolysis happens in the cytoplasm. So in terms of compartment, it's completely different from where the ETC is found, right? The ETC is part of the mitochondrial matrix, right? The inner leaflet. So that the NADH from the cytoplasm needs to be transported into the mitochondria. So that's where the malate aspartate shuttle comes into play, right? So basically, because the NADH produced from glycolysis cannot enter the ETC without the help of a shuttle, so what you're going to do is you're going to take NADH, you're going to donate that proton, those electrons in NADH to oxaloacetate, and you're going to convert it to malate. And it's the malate, right, which then is allowed into the matrix, right? Because malate is found in the Krebs cycle, in part, right? And then when you want to go the other way around, well, you convert aspartate, or you convert it into, I guess, glutamate to aspartate, and then you in total you have this malate aspartate shuttle. It's a little more complicated. Again, not super high yield. If your eyes are glazing over, you don't know what I'm talking about, that's okay. Don't worry about it. It's super low yield on the MCAT. Okay, make some note cards, flashcards on it, and move on. Okay. What is the glycerol 3-phosphate shuttle used for?
Is it going to make DHAP?
DHAP? Yeah, exactly right. Yeah, yep. So it's kind of a similar problem where the NADH can't, enter the mitochondria to participate in the electron transport chain, and then, like you mentioned, the NAD is going to donate its electrons to DHAP, the dehydroxyacetylene phosphate, right? That's one of these substrates in the glycolysis pathway, and it's going to form glycerol 3-phosphate. And that glycerol 3-phosphate is going to do, once it's, I believe, inside of the inner mitochondrial membrane, it's going to be converted back into DHAP. Okay, so it's very complex stuff. Not the most exciting question, I know. B is the correct answer, just like we found everybody picked, right? B is the shuttle that's going to be used to transport fatty acids from the inner membrane or through the inner mitochondrial membrane. And then A, the citrate acetyl CoA shuttle, what's that involved in? So let's think about this. Citrate and acetyl CoA. So acetyl CoA, where is that made at? Where's acetyl CoA made? Acetyl CoA is a byproduct of fatty acid oxidation, right? And fatty acid oxidation happens in the mitochondria, specifically in the mitochondrial matrix, right? And so when you have a situation, right? So it's inside the bacteria, it's inside the mitochondrial matrix where the acetyl CoA is produced. And if you have an energy-rich environment, okay, let's say you just had a really rich meal, and you want to produce more fatty acid, well, fatty acid synthesis occurs in the cytoplasm. But fatty acids are made of acetyl CoA, and acetyl CoA is found in the mitochondria. So the citrate acetyl CoA shuttle allows for acetyl CoA to be transported from the mitochondria to the cytoplasm to be turned into fatty acid. So if we want fatty acids to be synthesized, we pick A. If we want fatty acids to be oxidized, we're going to pick B, the carnitine shuttle. Okay, and they're the exact opposites. I could have probably done a better job explaining this. This is pretty complex. Okay, throw this on a note card, guys. Okay, I know it's a snooze fest. All right, next question. This one's fun. Formed by the liver during prolonged periods of fasting, which of the following are byproducts of increased fatty acid metabolism? Okay, so when you break down fatty acids, like diabetics do, right? Diabetics can't do what? Right? Diabetics typically have a insensitivity to insulin for a number of different reasons. Either they're resistant to it, or they have an autoimmune disorder, like in type 1 diabetes, right? So insulin's not really doing its job the way it's supposed to. And so the cells are never really able to take up a lot of sugar levels, or sugar, right? That's in the blood glucose can never take up blood glucose. So that's a problem. So the cell starts starving, right? They're craving energy. And so an alternative form of energy cells use are fatty acids. And a byproduct of fatty acid metabolism are ketone bodies. right? Ketone bodies. That's that fruity breath you may be aware of when someone has diabetes, right? You've heard of that before. So these acetone bodies produce that fruity breath. And the question basically asks, well, which of the four are ketone bodies? Are products or byproducts of fatty acid metabolism? So the answer choice isn't D, it's actually going to be C. So acetone is one of the three, acetoacetate is one, and 3-hydroxybutyrate is one, and then 4 is something I just made up. I just smashed essentially 1 and 2 together with 3 to come up with 4. So D is incorrect here, and it looks like a lot of people picked it. That's okay. I don't know if butyric acid is even a real thing. I'm sure if you Google it, it wouldn't come up. Okay. So that was a distractor answer that a lot of people picked. Baited. Baited. Okay. All right, so then we got 2 more questions left. Hang on tight, and then we'll do some psych-soc. Yay. Glycolysis, oxidative phosphorylation, and fatty acid synthesis occurs primarily in which cellular areas, respectively? Okay, so we talked about this, right? So this is me trying to reinforce what we just talked about, okay? Active listening skills. So glycolysis happens where? Andrew.
Cytoplasm.
Bingo. Nicole, where does oxidative phosphorylation happen?
Oxidative phosphorylation?
Yeah.
Mitochondria?
Mitochondria, right? Because endosymbiotic relationship, right? That primordial bacteria that used to, right? That was enslaved because they could do oxygen metabolism, right? Well, guess what? Anything that has oxygen, right? OxyClean, I don't know. It has to do with the mitochondria. Okay, simple as that. Boom. All right, so now we're between A and B. Cool. Now the question is, where does fatty acid synthesis occur? Lori, where does fatty acid synthesis occur?
Sorry, can I type it? Because I'm eating. Sorry.
Ooh.
Sorry.
That's all right. Yeah, type it.
Okay.
Hit me with that text. The suspense is killing me, Lori. Are you typing it? Okay, so Lori's got her mouth full and hands full. The answer is for fatty acid synthesis is cytoplasm, right? Because it's the mitochondria and the peroxisomes that do beta oxidation, right? Good. So the answer choice is going to be A. All right, and then the last question I had, open-ended question. Okay, and we'll probably draw this out. How many cycles of beta oxidation will be required to completely oxidize a 12-carbon fatty acid chain? And then the next question is, how many cycles for 17-carbon fatty acid? Okay. So we had a couple of people try this out last night. We had 6 and 7 and 6 and 8. And what do you guys think in chat? What do you guys think? Andrew says 6 and 8. What do you guys think? Anybody else? Go ahead and type your answer in real quick, and then we'll work it out. Work it out. And we're going to make it really simple. Okay, we're going to break this down so there's never any confusion. There's so many weird ways to rationalize this out. I've seen so many like shortcut tricks and stuff. They're just confusing. I'll show you guys the way to do it. Never get it wrong ever again. Help me out, Kev. I don't know. Yeah. Gave me a lot of grief too when I was studying this. All right, let's do it. So, when in doubt, draw it out. When in doubt, draw it out, okay? And that's exactly what you guys should be doing here. When in doubt, draw it out, okay? So, Trevor, 6 and 7? It is 6 and 7, yeah. So maybe I'll put a nice little 100% there. It is 6 and 7, let's talk about it. And maybe I'll do it down here. Fatty acid, beta oxidation. Let's draw that out. And maybe what I'll do is we'll copy this question onto here so you guys have the full notes for later. So let me do that. Discord, scroll up. Let me copy this question. And then we'll paste it onto here. $1 million question. Text, type, boom, copy, paste. Make this bigger. 15, bold. Let's make it even bigger. Shoot, why not? We got space, right? We're not restricted here. 22. Boom, bada bing, bada boom. All right, so let's draw this out. Okay. So my hope is that everybody can draw a fatty acid. If you can't, don't worry, I got you. So let's draw a fatty acid. Okay. So typically with a fatty acid, you're going to have some kind of ester group at the beginning, and then you'll have a variable number of tails. Actually, let me draw this. I guess this is fine. Yeah, we draw it this way. And in this case, we got how many carbons do we have? 12 carbons, right? So let's kind of count them real quick. So we've got 1, 2. So we got 2 right there. We got 2, 4, 6, 8, 10, and 12. Don't forget these first 2, right? The one that's on the carbonyl. Okay, that's where a lot of students make a mistake. And now let's connect the dots here, or the lines. Boom, 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 boom. Here's the trick. Draw it out. Okay, and all you're going to do is you're going to cut, right? Beta oxidation, one thing you know is beta oxidation is the removal of 2 carbons at a time, right? And that's going to be, those 2 carbons are going to be turned to acetyl CoA at some point. Acetyl CoA, right? So guess what? Slice it and dice it. So we got 1 cut, 2 cut, 3 cut, 4 cut, 5 cut. So how many, did I count this right? Hold on. 2, 4, 6, 8, 10, 12. Okay. So how many cuts do we have here? 1, 2, 3, 4, 5, and then I guess 6 right there if you want to do it that way. So we're going to have 6 beta oxidations. Or excuse me. What am I doing? It's 5. My goodness, guys. It's late. 5 for 12. Right? So when in doubt, draw it out. 1, 2, 3, 4, 5 red, not 6. 5 red cuts. How many cycles for a 17 carbon fatty acid? Well, I guess what? When in doubt, draw it out, right? When in doubt, draw it out. So that's what we're going to do. So now we're going to draw 17. So again, we got their little ester, right? And then we got 1, 2 already. 1, 2. So we got 2, 4, 2, 4, 6, 8, 10, 12, 14, 16, and then we got 1 extra carbon, right? So we're going to connect all that. Boom, 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 boom. Same thing as before. We're going to cut 2 carbons at a time, right? So maybe we'll do it this way. 1 cut, right? That's 1, 2. 2 cut, 3 cut, 4 cut, 5 cut, 6 cut, 7 cut. Okay, so you may be tempted to do an extra cut here, right? And that's the thing that's weird about odd numbered carbons, right? You actually want to maintain, if you have odd, here's a way to do it. If you start with odd chain, you want to end up with an odd carbon block, odd carbon block. In this case, it's going to be 3 carbons. This needs to be odd as well, right? 3 carbons for an odd chain. Okay. So how many cuts did we make for the 17? So this top one was 12 carbon. This is 17 carbon. How many cuts did we make? Well, 1, 2, 3, 4, 5, 6, and 7. So we have 7 cuts. Okay. 7 cuts for 17 carbons. What happens to this 3 carbon? That's weird. Well, oddly, this is going to be turned into, at some point, various steps, going to be turned into, I believe it's succinyl CoA. And then that will be fed into the Krebs cycle. Krebs cycle. Okay. Comments, questions, concerns? So it's 5 and 7. I think somebody said 6 and 7. Is that, yeah, you've been primed a little bit wrong there. I think I said that was right, but actually the answer is 5 and 7, not 6 and 7. Look at the Discord channel here. Yeah, so it's not 6 and 7, it's 5 and 7. Okay, so when in doubt, just draw it out. Just draw it out. Okay. When in doubt, draw it out. So I'll retract that 100%, Trevor. Okay. All right. Anything else on fatty acid synthesis before we can spend the next 30 minutes or so on some high yield psych-soc? Okay, I don't know which way to go. Everybody's quiet. I can call it quits here as well. And stop right here if you guys want. Oh, okay. All right. Cool. All right. Guess we're doing psych-soc then. Thought I was just talking to myself. All right, guys. Enough patronizing. Let's do some psych-soc.
Always here. I'm always here.
Always in spirit, physically, mentally, forever after. Okay, so typically when I do one-on-one tutoring with students, I do high-yield review the developmental stages, like Freud, Erickson, right? Erickson has all those different stages that people struggle with. Kohlberg, Piaget, okay, those are really important. And then if we have time, and Bandura, and then if we have time, I think we talk about some other stuff. I don't know if we'll have time tonight, but we can certainly continue it on next Tuesday if we don't finish. So let's do that. Let me do that. It's telling Spotify to click the wrong button. What happened? Guys, I need a new computer, so let's see here. All right, high yield psych-soc, here we go. PS high yield. Okay. Maybe Andrew could teach this to us. What do you think, Andrew?
No, I'm good.
Okay. I'm picking on Andrew a little bit because he is one of my one-on-one students, and we've gone over this, I think, before, right, Andrew?
I don't think we did psych-soc yet.
Oh, we haven't done psych-soc? All right, that's cool. All right, so let's do that then. So on the MCAT, right, in the psych-soc, a lot of students will come to me and say, hey, Kevin, psych-soc is something I'm really strong at because it's just definition-based. And I would agree it's more definition-based than the other sections, but it's not. That's a far cry from it being all definition-based, right? And typically, if you have that mentality, you'll probably get like a 127 to a 129. Let me qualify that. I got a 131 on psych-soc. And I was stuck at a 129 for a long time because I had that mentality as well. I'm like, oh, it's just definition. I'll be okay. I'll be good. Okay. But I couldn't improve my psych-soc score to save my life. I was stuck at that 129 for a long, long time. Oh, shucks. How sad, right? And so one of the things I did was when I was going through psych-soc, I really made it. What I did was with a study partner, you guys can do this as well. You don't need a tutor. You don't need me, for instance. You certainly don't need to pay me for that. But you can do it with among yourselves here in the study room. You guys can go through the answers. You don't have to just look at the question stem. Just look at the answers and make sure you can do two of things. You can verbalize an answer and you can provide a scenario, right? So for instance, Andrew, let me pick on you. let's say we were seeing a multiple choice on, let's see the word came up on a multiple choice that said representative heuristic. What is the representative heuristic?
It's the representative heuristic. Is it kind of like a bias?
Well, it's a bias, of course. Yeah.
You're making a decision based on, I guess, you think I'm not sure.
Yeah.
I can't really.
So I'm glad you're doing this, because a lot of students do this, right? They look at the word and they try to rationalize it out by looking at the word. And that's kind of wrong. Like, I understand where that comes from, but that's too stupid for the MCAT. Don't do that. Okay. Sometimes it's fine, but most often it's not. And a lot of students will say, "Oh, I know this word. I know it because I've studied it on my Anki. I've seen it a million times." And that's fine. But that doesn't demonstrate, you recognizing it on a flashcard is not the same thing as you knowing it. So I make all my students verbalize their answer choice, even if they think they know it, okay? Because it demonstrates mastery, right? Anki is kind of foundational. It's okay, but if you can truly talk about it and give a definition in your own words and provide an example, that's what you need to do for the MCAT. That promotes long-term memory, okay? So representative heuristic, does anybody want to give it a shot? Or maybe Andrew, if you want to hot potato to somebody else, call a friend. Pick on somebody.
Is representative heuristic? I mean, heuristic is a mental shortcut, or not, I guess. But then representative, is it the one where it's like something that's similar to the thing you're trying to remember? So something that represents. Shit, I'm using represent again, but it's a mental shortcut that uses. It's like it belongs in the same group. Like it's not exactly. Oh fuck! I can't. I know it.
Yeah, no, you don't. But you don't, right? You know, but
I don't.
But you don't. Okay.
You're gonna say it, and then I'm gonna say, yeah, that.
Anybody else? Last try. Come on, group.
I'll go.
All right.
Okay. So this is the one where there was like a question of the day a couple of days ago from Kaplan, and it was like, okay, this guy likes to go to orchestras and hear symphonies play music.
Yep.
Is he more likely to, like the representative heuristic would say, that will tend to believe that he played the trumpet growing up more than he played chess growing up? Correlates?
Kind of. Okay.
It seems to like fit into the category.
Yeah. Close. You're hot. Hot.
But it's not
That's what
there.
I said.
So very, very close. I like it. I like the brainwave on that. So the reason why I have students verbalize is to really make sure they understand it. And then if you can give an example, a scenario, it helps you kind of nuance or tease apart definitions that might be very similar in nature, right? You've probably seen similar words and you're like, wait a minute, what's the difference? Well, if you can give an example, it's going to help you remember that and understand the technicalities here. So let's so that's my example. So heuristic, representative heuristic essentially is this, right? Representative heuristic is when you see somebody, you think they're representative of a certain group. And this is also known as a stereotype, okay? So let's all imagine we just took the MCAT, we're partying it up, right? We're downtown LA or Miami, I don't know where we're at, right? We're downtown the city, drinking some brewskis, okay? Celebrating, summer out, it's nice, COVID's gone, okay? And we're outside chilling, and some guy comes up to us, and he looks like he's homeless, right? He looks homeless because his shirt is dirty. His jeans are dirty. He's got holes in his jeans. So we're like, dude, stay away from us. You're kind of gross. Yeah. When in reality, though, maybe he's not homeless, right? Maybe he's not that stereotype at all. In fact, maybe his car broke down. He's been in the hot sun working on his car, putting on a new tire, and he's been on his back on the cement, right? So we just employed the heuristic, the availability or representative heuristic, pardon. Let me give you another example. Let's say we're actually in the bar, right? Maybe it's just like you and I don't know, having a glass of wine, BSing a little bit. And white Urkel, right? Urkel walks in, birth control glasses, suspenders that don't fit right, tube long socks on. He walks in and we're looking at each other, crack a smile and say, man, that guy's a dork. When in fact, he'd be the coolest cat in town, right? Maybe he's just got an amazing personality. He's just down to earth. Okay. So is he a dork now? Maybe not. Okay. But we employed the representative heuristic because that's what we associate. What he's wearing is representative of what we think a dork looks like or a homeless person looks like. Okay. Cool. What about the availability heuristic? Anybody want to give that one a shot? Availability heuristic. What is it? Carolyn, hit me up.
Okay, so this is the one where you're like basing the probability of something happening to you at like how quickly it comes to your mind. So like, I think the classic scenario that I first learned was like, you see that there's a lot of like car burglaries happening. And then you go outside and you start to think like, oh, well, my car is going to be stolen soon. Like I better lock it.
Right. Good. Yeah. So let me give you another example. I like that. Perfect. That's exactly what you guys should do for all these terms that you see in Psych-Soc. Don't worry about what the question stem says. Just go through those question and answer choices and make sure you do that for most of those, right? So let me give you an example of the availability heuristic as well, right? So the definition I would give is that when you place more emphasis on a piece of information that you most recently heard, right? The most available or most recent information. And so a scenario might be this. Let's imagine downtown LA again. Okay. We got the top down in the car. We're cruising, right? Our hair's getting caught in the wind. We're getting kissed by the sun, right? We're headed to the beach, all right? Having a good time, having some laughs, looking forward to it. And we turn on the radio and it says, breaking news. Breaking news. A man from New York just got bit by a shark. We're like, oh, shit. Now we can't go to the beach. We can't have a good time. We can't go body surfing. My plans are ruined. Maybe we shouldn't go to the beach now. When in reality, what? When in reality, one, it's in New York. New York's a long ways from California. Even if it was in California, what's the likelihood that we're going to be bitten by a shark? Probably slim to none. In reality, it's more likely we're going to get in a car crash traveling to the beach. But we're placing a stronger emphasis on the most available information to us, right? So that would be an example of the availability heuristic. So again, make sure you guys, some sage advice, make sure that you can do that for every term for Psych-Soc.
Kevin, can I say something?
One of the other things I did, sure.
So that first example you gave of like the homeless guy, I've been thinking about it ever since. And I think, wouldn't that be better classified as like fundamental attribution error? Because that's why I get confused with that one.
Well, attribution error is when somebody does something stupid and you typically want to say that the issue is something inherent versus situational. So it's related. It could be, yeah.
Okay,
yeah,
sorry. Keep going. Never mind.
Yeah, right. Maybe if you see him, you think he's stupid because he's homeless, right? He's homeless because he's lazy. That would be more fundamental attribution, right? When in reality, like I said, it could be an external event, right? Maybe his car just broke down. Maybe he ran over a nail. I don't know. Okay. It's related.
Thank you.
I like it. Yeah, I like it. So the other thing that helped me go from a 129 to 131 is not just doing that with a study buddy. Okay, so guys, please use these study rooms throughout the week and just quiz yourselves on these terms, right? Go through an old test together with somebody and go through it just like we're doing now. In addition to the thing that helped me was to do the Psych-Soc on New World. Okay, Psych-Soc on New World is pretty good. I know a lot of students speak highly of UWorld, and I do too, but I think in particular their Psych-Soc is very good. I don't like UWorld for cars. I think the best place to do cars, for instance, is AAMC, but that's for another time, another discussion. So if you do that, one-on-one students where I say, hey, for every 4 Psych-Soc sections we do together on a test, 4 tests, Psych-Soc sections, your score should improve at least a point or more if you do all those things, whether we do it together or not. A point doesn't sound like a lot, but it is. Okay. So if you've already taken 4 practice tests, we'll go back and review the Psych-Soc in this manner. Anyway, let's talk about some developmental stages. So when you do Psych-Soc, right, the first paragraph is typically intro. The second paragraph is study design. So you might see in the second paragraph in Psych Soc the following. It's going to let me write. There we go. We measured, right, X response in let's say in participants between the ages of 9 to 12 years. 9 to 12, I don't know, something like that, right? You guys have probably seen this before. And when you see ages being used, you should automatically be cued to developmental stages. That is a telltale sign that you're going to be dealing with developmental theory. You don't know which of the 4 you're going to get, but you're going to get at least one guaranteed. So should you know developmental theories? Heck yeah, you should. That's easy giving points, right? So the first one you should know is Erickson stages of development. And let me paste that. Boom, here it is. It's almost like I have this prepped. Okay? And this is gross, right? I'm guessing some of you have already tried to memorize this chart, right? Saying some nods possibly, right? Up and down, and maybe you've gone to it multiple times now. Trying to remember the order of this. And maybe you've gone to Google and tried to find a mnemonic. I think there's one out there that says something like trust auto in industry something. I can never remember that one. Okay. Here's how I remember this. And ladies, I apologize in advance. It's another perverse one. The way I remember this is this mnemonic. My sexy girl, or I guess you could use guy, sexy girl or guy, okay, making it EO. In red is sucking the D, sucking the dumplings. Sucking the dumplings. Another perverse one, right? Perversions kind of work. My sexy girl in red is sucking dumplings. My sexy guy in red is sucking dumplings. So my is for mistrust. Sexy is for shame. Girl or guy is for guilt. In, I guess, is for in or industry, inferiority or industry. Red, oh, there's an error in this thing. This should actually be role confusion. Role confusion, not just confusion. Red is for role. Is for isolation. Sucking is for stagnation. Dumplings are for despair. My sexy girl in red is sucking dumplings. I love me some dumplings, all right? I'm a foodie. No shame in my game. Okay. But this should help you remember the order. And that's a lot easier to remember than trying to rote memorize all these different stages in order. Okay. And you will need to know them from the MCAT in the order. Now, what about the age groups? Well, yeah, you know the age groups, but I think by knowing the chronological order through this mnemonic, my sexy girl in red is sucking dumplings, that shouldn't be as much of a problem. In fact, I think the only age group that you really need to memorize is this 13 and 21 down here. Everything else, ignore. Everything else you can rationalize, I think, based on the order of these stages. Now, 13 and 21, I think, should be very obvious in terms of identity and role confusion. We've all gone through it, hopefully. Most of us have, anyway. But middle school and high school, what do you remember from middle school and high school? Well, you remember like groups, right? There was the jocks, the nerds, the band geeks, right? Other types of people, goths, right? Maybe you're I don't know, experimenting with your sister's makeup. I don't know. Do you? And then if you go into college, guess what? You're trying to kind of reinvent yourself again, right? You're trying to reinvent yourself and kind of figure out who you are as an individual. So maybe, you put down or you put down your sister's makeup, no longer are a goth. Cool. Do you? And now maybe you're hanging out with the nerds, or maybe maybe you got into basketball. I don't know. Point is that you're continuing to identify who you are in college, right? You do a little bit of experimentation, yeah? And then towards the latter end, for most people anyway, latter end of college, okay? What are you trying to find? Well, you're trying to find, right? Once you've known yourself, once you've learned to love yourself or understand who you are, you're ready to accept somebody else in your life. That doesn't always happen for everyone, right? But approximately 13, 21, after 21 years old, give or take, right? Afterwards, you try to find long-term relationships. Intimacy, right? So 21 plus, you're trying to find intimacy, right? Trying to find your partner, trying to settle down a little bit. I don't know. buy that house on the hill, right, with the white fence. And if you're a pre-med student, you probably already have your parents or your grandparents asking when you're going to have kids. All right. So this should be very natural. You should understand these stages, I think, just fine. And hopefully, this perverted mnemonic that I shared with you guys, it'll help you. Again, I'm not making any apologies for this mnemonic. It works. Makes it a lot easier too.
Sorry, I have a question.
Okay.
So is this the correct age? Because on a diagram that I have, it says 12 to 18 for identity and role confusion, and then the next one is 18 to 40. But this one is
no.
Yeah.
So okay, I see what you're saying. Oh, I see. Okay, yeah, it's fine. Every resource can be slightly different from like a couple ages. Not that big a deal.
Okay.
They're approximate. That's all it is. Okay, so don't try to hard memorize these age groups. Really. Okay, use a little bit of logic, common sense. You'll be fine. Okay, great question. Yeah, and we'll see that for PJ too, where some sources will go from 0 to 2 and then 3 to 7. Other sources will do 2 to 6. It's okay. If you're off by a year, it's not going to be the end of the world for the MCAT. It's not going to be that picky. Okay, it's going to be very obvious what they're asking you. Okay, there's not going to be any gray area or some weird boundary difference. Okay, yeah. All right, so my sexy girl in red is sucking dumplings, or my sexy guy in red, A, is sucking dumplings. All right, keeping it spicy. I like it. Let's talk about the next one. PJ. We're going to draw PJ out. Let's scroll down. PJ. Where's my stylus? There it is. Cool. PJ. PJ. You know this guy too. And the first stage is from 0 to 2 years. PJ, you do need to know the age groups. Okay. First one is 0 to 2 years. Jesus, can't type. 0 to 2 years. What's the first stage called?
Somatosensory.
Sensory motor.
Sensory.
No worries. Sensory motor. I know what you meant. And what is the defining feature of sensory children, or I guess infants, develop in this stage? They develop something called object permanence. And what is object permanence?
I think it's the idea that like you start to understand that even if you can't see the object still exists.
Perfect. Chef's kiss. Mwah! Perfect. Perfection. Exactly. Okay. So object permanence means that the child understands that even if he can't see the object, it still exists, right? So when you first have a child or infant or whatever, right, they will be amazed by peekaboo, right? When you hide your face and do peekaboo, they'll be amazed because they're like, man, you just reappeared and disappeared and then reappeared. It's magic. That's exciting. That's fun. But they're going to pick up on that pretty quick. And now peekaboo is not going to be that exciting because they developed object permanence. They know your face still exists behind your hands, or that toy still exists even if they can't see it. Right? That's not fun. Next stage is from 3 to 7. You just slap 4. Okay. So this one is pre-operational. Pre-operational or pre-op. Well, maybe not pre-op because that's something different. I don't think too many kids are undergoing sex changes, but pre-operational, right? Pre-operational. And this is defined by egocentrism, role play, and imagination. Let's talk about these in turn. Role play, right? This is, remember, like, I don't know, when you were little, kindergarten, maybe first grade, okay? What did you do in first grade in recess? Well, maybe you thought you were a Power Ranger, right? Maybe you were the baddest, bam, 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 Pink Ranger or Green Ranger. I don't know, doing some karate, right? Or maybe you were Mommy Daddy, or maybe you were, I don't know, Cops and Robbers. I don't know. Maybe you were John Elway and playing QB for, football, okay? Imagination. At this age, right, a lot of kids have wild imaginations. Maybe they think they can fly. All right. Well, R. Kelly song, I Believe I Can Fly, right? Or maybe you thought you had the boogeyman was living underneath your bed, or maybe in your closet, right? So you hid underneath your blanket because your blanket was a protection zone, and you made sure not to have your feet exposed because they would eat your feet, your toes off, right? And then you held your teddy bear tight, right? Egocentrism. Let's talk about that. It's like a Toby Keith song. I want to talk about me. I want to talk about I want to talk about number one. Oh my, what I think, what I like, what I want, what I see. It's very true for children 3 to 7 years of age. And you've experienced this yourself. You've probably gone to a girlfriend's house who has a kid, or a brother or sister who has children, to their birthday party. Or maybe gone to the park and witnessed this third hand. And a little kid goes up to his mom screaming and crying and being all mad. And the kid goes, Mommy, I want to play with that toy. That's my toy. I want to play with it. And the mom says, well, little Timmy, you need to learn how to share. You only want to play with that toy because the other kid took interest. Right? Egocentrism. Cool. Next one. So 0 to 2 is where you start off. You slot 4. The next one, you just slot 4 again. So now it's going to be 8 to 12 years. 0, 2, 4, 4. And the next one is what? What stage is this? If we have pre-operational, now we're going to have concrete operational. And what is the defining stage of concrete operational?